So every students, please write your name and UID in the chat box. Okay. Today, respected HOD sir will take class on flow cytometry, and so try to open your cameras by 10 o'clock or within 5 minutes. Make yourself comfortable and open your cameras. Okay. Thank you, Keshav.
Good morning, sir.
Yes.
Good morning, everyone.
Yes. If possible, please turn on the cameras within 5 minutes.
Now screen is showing or not?
Sir, your screen is not at shared, sir. It's not showing.
Not sure. Huh?
Advanced sharing. So I made you as co-host. Please try now, sir. Sorry, sir. I made you as co-host. Now you can able to share, I guess.
Okay.
Okay, very good, Mukhtar. So today HOD sir will take a class on flow cytometry. Please wait, students.
Still not sure. Huh? Something. Can you send you can you share from your end?
Yes, sir. I can. 1 second, sir. I'm coming for this.
What is the issue, sir? Recording in progress. Where is the once go to Zoom, sir. Okay. Once. Please screen. Then Zoom. Share. File, na? Basic. Okay. PowerPoint. Share. That's it. It's okay now. Thank you so much. Hello, everyone. My voice is clear?
Yes, sir. Yes, sir. Voice is clear. And your slides are visible.
Okay, thank you so much. So today we will discuss about the flow cytometry. So all of you knows this is the advanced technology like auto analyzer, we are hematology, hemocytometer we are using. So just this is advancement of flow cytometer. Slightly the advanced, the principle is just basic principle is same, but the advanced principle we use some stain, some separate, not like the cytometry. And so that's why we will discuss today flow cytometry. What is the basic principle? You know, the hemocytometry, the impedance technology, the optic technology, same thing, the same principle working here. Only the one important thing here is the flow cell, and they have the laser light. So laser light is the making the flow a single cell movement. So this is the one of the important things. So the single cell you can analyze in the different way. So how different way? So we want to see the single cell. So not only the count here in the flow cytometry, we want to know the structure, their cell surface, their nature, based on whatever we have the cell on the we are identifying. So not only the nucleus, not only the granules, the cellular, the surface of the markers are also present, then we are identifying. So the saying, the amount of light scattered at the right angle to the incident light beam depend on the internal complexity of the particles. So we know that the wide angle of the side scattered and the side scattered detect of 90 to the laser beam. Why important the side scattering in the cells? Because when the light pass through the side, then the cellular structure you can see the very easily. So that's the side scatter channels are the important. Okay. So how the 90-degree light is scattered. So the 90-degree light is scattered. 1 minute, just hello. Yeah, good morning. Yeah, please, yes, sir. I'm class. Can you give me the time? Thank you. Sorry. So the laser light, they move the 90-degree angles. So the sensors, they can take the all areas, means the whole cells, you can rotate and you can identify the easily. So this is done by the through the optic, like same principles, they are using the optic. So what are the properties of the forward scattering and side scattering? So there are two things, forward and side. So we can get the with the 90 more than the 90-degree angle, the side is scattered and the forward scattered. If you are checking, so this is the principle. When the light pass through the particle or cell, anything you can say, so that the both angles. So one is the forward. You can look at the picture. So forward arrow and the side scattered. So side. So both angle we can whole cells we can identify. So how in the forward scattered? So diffract the light, so related to the cell surface area, so that you can see the surface area. Then detect along axis of incident light in the forward direction. Okay, this is the forward scatter. Second, side scatter. What is side scatter? Side scatter is reflected and refracted light. And second, related to the cell granularity and complexity, detect to 90-degree to the laser beam. So any surface, whole cell surface area, if you find anything, then you can identify. So basically, this is the optic principle for this. Now the same principle in the auto analyzer like 5-part differential are also doing, no doubt. But what is difference? Then we will discuss slightly the next. So all the cells passes through the laser beam. So what is the properties of this? So as the cell passes through the laser beam, so light is scattered in all direction, and that is scattered into the forward direction and proportional to the square of the radius of the shape and is to the size of the cells of particles. So because of in the light, so easily they can the movement. So if any activity, any area, any things, so through the laser beam, we can easily identify based on their area, based on their radius, based on their size, and any particles are present inside the cells or even the surface on the cell. So the cells may be labeled with the fluorochrome linked. Now, so what is that one things in the flow cytometry? We are using the color, fluorescence color. So many different we will discuss in later, because today is just the introductory class for flow cytometry. Later, I will show you the detail, because flow cytometry is not the one-day lecture. We have to take the 2-3 lectures, but I will try to compile. So when you the fluorochrome, fluoro means fluorescence, color means color. So fluorescence color linked with the antibodies, to stain the fluorescent membrane and cytoplasmic and nuclear dye. So based on this, we can easily identify if there are anything present on the cell surface. Second, the cellular identification by flow cytometry. How cell identified? So the first is its relative size, means relative size is based on the forward scattered cells. So forward scattered cytometry, we are saying the FSC. So this is the relative size. Second, side. So side scatter or SSC, you can say it is related to the granularity or internal complexity. Means in the cell present the granules or nucleus or nucleoli, anything present inside, so that we can identify. Third one, so see the first and second, this is working in the latest hemocytometer also, no doubt. The third one is the only found the flow cytometry. Its relative fluorescence intensity, like 1, 2, 3, 4, 5 channels. Nowadays, 1 channel, 2 channel, 3 channel, 4 channel we have. So multi-channel flow cytometry. So we are different companies are allowing, like Guava, they are giving just 2 parameters. So as per your requirement of the labs, like you know the only CD4, CD8. Some lab, HIV lab need only CD4, CD8. So government has by only the this analyzer. But if you are working in the stem cell lab, then you need to the multi-channel or 4-channel. So based on that, we are using. Now, what happened in the optic fluorescence channels? So any fluorescent molecule present in or the optic on the particles will absorb energy from the laser light and release the absorbed energy at longer wavelength. Okay, the emit light is collected by lens and detector. Already detector means the the one of the computerized, through the computerized they detect, and emitted fluorescence intensity to proportional to the amount of fluorescence compound on the particles. So based on how much they present and according to the proportional things they are counting. So this machine they just get the data, collect, detect, and then through the software interpreted. So the working in the software also one of the important things, how you work on the software. So for how you are cell sorting. So these are the things we have to know. This is the need to the expertise. So basically here in the fluorescence, you understand this fluorescent light, this color dye especially, this bind with the molecules. And once the molecule bind, then the laser light if come through this, pass through this cellular or stain cells or particles, then they detect based on their wavelength. Because of the color, whenever the color come, you need to the wavelength. So some flow cytometry, they're taking the automatic wavelength, or some you have to fix the wavelength what. So already we have the program, or you can create the program based on your requirement, right? Clear this point? Okay. So
Yes, sir.
how detect? So once the laser light see pass, then through the flow cell, the cells is passing. So the fluorescence different color. Suppose this granules all granules have different in nature. So based on their color, they detect and then make the plot, the graph, and based on graph you can identify. Okay. Now the optical design. So optical design. Okay. So how it's working in the optical design, just look at here. This is the chamber. This is the chamber. So in chamber, when you shape this, so before how we are doing the processing, so we are taking the blood amount, whole blood or stem cell or anything, and you have to keep in the fit C, we are saying that a specific color, a specific fluorescent color, you have to take it, choose your according to your, some for the viability test, some for the CD34, CD45, according to the requirement, you are choosing the cell. Then you are putting the proper in the, as per the manual, or you are keeping in the incubator. Once the incubate, then you have to process. Once they start the machine process, then flow cell will start, then take here, and then the samples come in the light scattered sensor. Through the sensor, then they reflect. Here the sample pass in the detector, in the flow cell, then start. So what happened here? Through the cell, you can see the brand pass the filter because of the light. See, even the light comes, so light is reflecting. So this light through the dichrome filter, means they have the different color filters. Through the filters, dye means two colors pass through, and then they detect the individually area. So one detect, and then give you the make, suppose RBC, WBC, platelets, or different, even the, if I talking about the stem cell, then CD34, CD45, they have different, and based on they detect and then make the graph and the amount. So when you, during the analysis, I can show you, based on this, you can, machine already they give the how much cells are present. Clear this graph? This picture is clear? Okay.
Yes, sir.
Okay. Yeah, please, because I don't know voice is, please just respond me. Third one is electronic part. So the scattered light from the particles passing the laser light is converted to digital value that is stored in the computer for analysis. See, electronics, you know, the important part means through the, whatever the light, this through the detector to the software in the system, the electronic role is very important. So whatever the scattered light passing through the particles, so laser light is converted to the digital value. Understand this point? Laser light converted into the digital value. So how much this light passes, because the light passes through the filter. So based on, they converted the value in digital, and this is the store through the software. This happened through the software. So laser light converted to the digital value. How much? Quantitative. And then store in the computer for analysis. So this time is stored. So you can store analysis, you can do the later. Once the store come, you can do it. And once the free, what is the in the flow cytometry system? So once the cell sorting is, first process is cell sorting. Once the cell sorting finished, process finished, then you have to go for the analysis. So later on, you can sit and then you can analyze that data. So this is the once, see, the one the flow cell count means appear, then come to the detector, and detector to the amplifier. Amplifier, you know that here is the procedure to the converting to the light to the digital value, and then come to the ADC, where the computerized have a system converted through the software or your through this stored in the computer system. So this is the basic principle of the working principle of the flow cytometer. Now, how the like already we discussed in the side scattering, how the granules or intracellular structure they are finding. So based on the relative size. So forward scattering, you are looking the size, and the side scattering, you are looking the structure of the internal or external. So internal, like granules, like nucleus or anything. So granularity, because every cells have the granules. So how much granularity, thick, light, minor, whatever. Then resonance or relative, how much they have. So based on that, so example, this is the leukocytes. So leukocytes, you know that they have the granules, they have the cellularity, and then the resonance also. So based on see, if the color comes, so how much leukocytes? So if the more, so go for the above. If the less, so see the this green color less. So less than 50,000. This is the different cells, the 100,000, and then leukocytes is very, very high. So in the graph, you can identify, because the light through convert in the digital at this work as a counting number value. Now, then the gating. What is the flow cytometry? The basic things, gating means gathering, whatever. So gating is the essence electronic window that set upper and lower limit on the type of amount of materials thus pass through. So how much? This is also important. Sometimes very few things are in the also identified by the flow cytometry. So it is used to separate a subpopulation from the heterogeneous population. Means separation. You know, the subpopulation means if the example progenitor cells. So progenitor cells are also, you can see, from the heterogeneous population. Heterogeneous means different type of cells. So any type of different cells, you can easily separate, you know, this is the from this series. So that's why we are saying we are doing the phenotyping. So phenotyping is the flow cytometry main, the USP is we are doing phenotyping. So it permits very specific questions to the ask about the particular population. So this is the identify. You can easily say, oh, this is belongs to the particular this cells. Now, based on this, you can see the fault. The granulocytes, the number one have the more, then monocytes, and third is lymphocytes. So based on the presence, they make the graph. The next, the fluorescence and fluorochrome that already we discussed, because this is the important things. This is the reason we identifying the phenotyping we are doing. The coupling of monoclonal antibodies with the fluorescence dye is necessary for the recognition and enumeration of by the flow cytometer. So need antibodies. So monoclonal means single. You know the monoclonal and polyclonal, I think everyone. Why we use monoclonal here, not polyclonal? Because we need the single, specific. That's why we are using monoclonal.
I didn't get that. Could you try again?
Okay. So each fluorochrome passes a distinctive spectrum pattern of absorption and emission. So two things here. One is absorption and second is emission. What is absorption? Any color substance, the light has nature to absorb, that you know. Also another we can see the excitation, and emission means how much emit. So the property of fluorescence is that the fluorochrome present on the cell absorb the light, laser light, and re-emit the light at a lower energy and longer wavelength. So this is the one of the basic principle of these things. The most popular fluorochrome used in the immunofluorescent is fluorescein isothiocyanate, or FITC we are saying, which has an absorption maximum between 50 to 550 nanometer. So, you know, this is the FITC color, this fluorescent dye. Actually, this is the very, very common use for the any flow cytometry. And why? Because their color power, the 40 to 550 nanometer wavelength, they can easily absorb. So the FITC emit light detect in FL1 on the FCM flow cytometry. So this is the one, the dye. So this is the fluorescence emitted dye. Another example of the excellent combination is the phycoerythrin, or PE, as a second fluorochrome dye. It can absorb light at higher wavelength than the FITC. So according to what your need or what you want, then you can change your FITC, or you can use the FITC. If you need only the cell viability and other basic structure, you can go with the FITC. But if you want to go for the or internal things, then you can go for the PE or phycoerythrin. So phycoerythrin emit light can be detected in FL2 on the flow cytometry. Okay. So not only we have more than 4 or 5 channels we are using. Okay. So detailed things we will discuss how we procedure. Now, What are the application? This is the important. Of course, immunophenotyping. We are here writing for the HIV. We are usually using, but it's not the only HIV. We all immunophenotyping we can do. What is the immunophenotyping? Anybody knows? Phenotyping. Have you understand about the phenotyping or genotyping? Are you there? Anybody? My voice.
Yes,
My voice
sir.
is. Yeah, I'm asking. You know the phenotyping? What is phenotyping? And what is genotyping?
Like
Your
XY
voice is breaking.
chromosome, like XX, and physical like how physical we like have a human have a, like a brown eye color, that is phenotype.
Hello.
The genotyping is analyzing the gene composition of the cells.
Right. Good. And phenotyping? Yes. Your voice is clear.
I'm not sure about phenotyping, sir.
Okay. So phenotyping same like you are analyzing the gene or the chromosome or genetic area for genotyping. Cell surface, we analyzing the cell surface or cellular level or internal or external, we are saying the phenotyping. Okay. So when we discuss the immunophenotyping, that means this with antigen, antibody or anything present on the cell surface. So that's why. So markers, basically the any type of markers, how we identifying the stem cell, how we identify the progenitor cells. So they are nature, because stem cells have only the markers you can identify. So that we have the more than 350 around the CD markers, cluster determination, this said. So we all we can find out through the flow cytometry, but you need to change your color or your requirement, your channel. So first we are using the immunophenotyping or phenotyping for the CD4, CD8 count. You know that. Leukemia and lymphoma immunophenotyping. So based on what type of present. Cell cycle and the plody analysis of tumors, we are identifying based on their marker present on the surface. Reticulocyte enumeration, because sometimes if you want to know the details of reticulocyte, what type of reticulocytes, then you can enumerate. Now I'm in meeting. And second, flow cross matching, the organ transplant, stem cell enumeration, you can detail go for the stem cells, residual white cells detections, and quality platelets and red blood cells count, you can do it. Second, research laboratory you can use as a immune functions studies. Means I wrote the broad, you have to go for the detailed things. Hematopoietic stem cells, multi-drug resistant studies in the cancer patient, kinetic studies like cell function, platelet analysis, coronary disease, environmental sample analysis, flow and the the in situ fluorescent in situ hybridization. Okay, so these are the things you can use as a through the flow cytometry. So in the immunophenotyping, you can use by the cluster determination. Already I told you through the using the single monoclonal antibodies, or you can identify. So the cluster determination or cluster designation is group of antibodies that all recognize the same antigen. So the performed by labeling by the cells and red, green, and the orange labeled monoclonal antibodies. The cells are the interrogated in the flow cytometer, and the percentage of positive cells are recorded. Okay, so both things. By positive, so positive, negative, we are when you are study the phenotyping, then based on there you can do it. Now I'm going slightly fast because the 8 minutes is left. So if you anything, please let me know. So these are the some markers for the T cell markers. This is the myeloid cells markers. So we have the B cells markers. So based on markers, we have the panel. So now there any panel you can use. So CD2, CD3, 4, 5, 6, 7. They are the T cell myeloid markers like CD11B, 11C, 13, 14, and 33. B cells markers CD10 to 25 or the SIgG, IgM, kappa, lambda. Miscellaneous marker like HLA, DR, DQ, CD34, CD45 for the stem cell, TDT, CIgM, and C complement D3. So these are the markers we are identifying through the immunophenotyping. So now some types of instrument already discussed. So some the part have the intrinsic parameter like they are giving the cell size and granularity. Extrinsic parameters like cell surface antigens, they're single or multiple surface membrane. Antigens are detected with the fluorescinated monoclonal antibodies. Example CD45, CD4, CD8, etc. So many things. I mean, all you can do it. Another, you have the multi-parametric flow cytometer. In multi-parametric flow cytometer, we are using for the measurement of such 3-color surface cytoplasmic stain or conjugation with the cellular DNA content. So for the leukemia and lymphoma, you can distinguish myeloid leukemia from the lymphoid can be precisely determined by the analysis and surface of cytoplasmic antigen. So you can easily identify difference between the leukemia and lymphoma. Also, others use the flow cytometer as for the platelets. You can use for the Glenn-Jamin thrombocythemia, Bernard disease, stored pool disease, reticulated platelets, antiplatelet antibodies, and monitor treatment with the Glenn-Jamin thrombocytopenia or thrombostasis. So for this purpose, we can use the flow cytometry. DNA content and cell cycle analysis. The aneuploid pi or elevated the S-shape function have been shown to be the prognostic indicators in breast cancer, colon, rectal, prostate, and bladder tumors. So any tumor analysis we can do through the flow cytometry. So these are the normal cell cycles we can identify for the DNA contents. Now we can just, the G grade, S grade, we have the 4 cell cycle. So then we can easily identify through the how much they have this type of color. So based on the color, suppose this CD45, so you have to sort in. Here you can just collect, you have to manually, you can select it, area, then automatically you can say that this much. So count will become. So this is the part of short sorting, and we will discuss later on how we have to use. So this is the side scattering for the CD14, CD45. So just you have to click here like this, and you have to select the area. Automatically the count will be showing. So now we have to check it. So as per your experience, your expertise, because of one cell, if you are capturing, then your result will be very, very varied. So here we need to the expertise based on, so because different colors, so easily you can do some area here, this type of the mixed color coming. So that you have to identify which cells we have to take it. So this is the based on your experience will come. Okay, guys. So this is the basic flow cytometry. Detailed procedures and things we have to discuss more and more. Cell sorting is the big things. How can use the software? That also is the one of the important part. We will discuss later on. So anybody, if you have any basic things about the flow cytometry, please ask. We have 4-minute time is left. Yeah, one by one.
Yes, students, if you have any queries, please ask, sir. And thank you very much, sir. The class is very interesting.
Thank you.
Of flow cytometry and its applications. Is there any queries, students, regarding flow cytometry?
No, sir.
No,
No?
sir.
Okay. Okay. Actually, we already finished this flow cytometry, sir. Already students are clear with it about
Okay.
forward scatter,
great.
side scatter applications. So
Yes. Actually, that's why today first lecture, so I just choose the basic things because I
Exactly.
not capture the analysis part. because the cell sorting is the one of the big topic, and then how can use. So we can capture another lecture.
Yes, sir. Okay.
Okay, thank you.
Okay. Then we can finish this. Actually, only 3 minutes left. Okay. Thank you very much, students. You can leave.
Okay, thank you so much.
Yes, sir. Yes, sir.
Thank you very much.
Thank you very much. Everyone can leave, students, now.
Thank you.